One of the interesting things that occurred in the 1880s was that due to Koch's postulates, a lot of scientists, early microbiologists, were trying to develop pure cultures. One of Koch's postulates says that you have to start with a pure culture. And so this was really difficult to do in the 1880s. The only way you could grow bacteria was to grow them in broth. And if you got one other bacteria in there with your bacteria, they would become contaminated, and you couldn't use them to prove Koch's postulates. So in the early 1880s, his lab tried to use gelatin. They tried adding gelatin, which is a protein derived from animals, to put into the broth to make it kind of semi-solid. There's a problem with using gelatin. First of all, when you warm it up in an incubator, it gets more runny. It doesn't stay solid. It only stays solid at cold temperatures. Think about Jell-O. That's an example of gelatin. The second thing that's a problem is that many bacteria produce enzymes that can break down proteins. So if you tried to isolate a bacteria that produced a potent protease, all your gelatin would turn to soup, and it was a big running mess. So it didn't work very well. In 1882, the wife of one of the scientists in his lab, Frau Hess, was making jelly, and she was using a different substance called agar. It came from a seaweed, and she was using this material to solidify her jellies. And she suggested to her husband, why don't you try using agar? Agar was a great substrate. It comes from a red seaweed. It is a polysaccharide instead of a protein. And if you heat it up and melt it and let it cool down to room temperature, you can warm it up to 40 or 50 degrees and it stays solid. It doesn't get soft and runny. But in addition, most organisms that they were working with in the lab have never seen this seaweed before. Therefore, they had not developed any enzymes to digest the carbohydrate. And therefore, you didn't have to worry about proteases like you did with the gelatin. Shortly after that, another gentleman, Petrie, proposed growing, instead of growing organisms in a flask, he proposed growing them in modified dishes. The dishes look like this. It was one large round disc setting over a smaller round dish underneath it. And if you sterilize the dish and kept the lid on, the media inside would not become contaminated. The side of the dish worked much like Pasteur's S-shaped flask, in that it allowed air to go up the side and over the top, so air could get inside the dish, but any dust and contamination would be trapped on the outer lip of the outside dish and could not get into the interior to contaminate the media. This is known as the Petri dish, and we still use these today. Theirs were made of glass. Today, ours are made of plastic. They come in a lot of different sizes. Some of them are split in half, so you have biplates. This is Theodore Eicher. We mentioned him earlier in the lecture. He was a pediatrician, and the interesting story about Theodore Eicher is that he discovered an intestinal bacteria that aids in infant digestion and health. This bacteria later on turned out to be important for all human health. He studied this bacteria in the 1880s and associated it with problems that some infants were having with digestive diseases. Theodore Eicher wasn't recognized at that time for the monumental contributions that he had made. It wasn't until 1919 that, when scientists were reviewing his work, discovered how much information he had provided about this important digestive bacterium. And it was in 1919 that the scientific community decided to change the name of the organism in his honor, and therefore it was named after him, and that's where we got Escherichia coli, or E. coli. It is the most studied bacteria of all. scientists knew that you could induce immunity into animals and humans, but they didn't understand how it worked. There were two theories. One of these was proposed by a Russian scientist named Mechnikov in 1884, and he had been working with starfish larvae and noticed that these larvae could engulf bacteria in solution, bacteria and algae and smaller microbes. And he called this process phagocytosis. Later on, he was looking at white blood cells, and he noticed that some of the white blood cells could do the same thing. That if you put bacteria in a solution with white blood cells, that the white blood cells would phagocytize the bacteria. And light bulbs went off. He said, aha, this is the way that organisms protect themselves. This is the way they develop immunity. It's through their white blood cells. And he called this theory the cellular theory of immunity. And here's Mechnikov, or a cartoon of him. Here's a starfish larva. And there's a white blood cell, or a phagocyte. But that wasn't the only theory that was around. Another gentleman named Paul Ehrlich had a different theory. He was able to take blood, filter out all the cells, and use this serum to pass from animals that had been immunized to naive or unimmunized animals and protect them. So he said, no, it's not the white blood cells. There's some mysterious chemical in the blood that is responsible for immunity. And he proposed the contradictory theory, which was called the humoral theory of immunity, or better today we know it's due to antibodies. These two scientists fought like cats and dogs. And it turns out, in the end, both were correct. The immune system is composed of two parts. One of these is cellular, and one of these is humoral. And they both work cooperatively to give us and animals immunity to diseases once we've been exposed or vaccinated. Both of these gentlemen were awarded the Nobel Prize in 1908 for their theories, which formed the foundation of immunology. There were many diseases that were caused by agents which could not be visualized by microscopes, like bacteria. So they call these agents viruses. However, up until the 1890s, no one had been able to purify a virus or to isolate a virus, and so the first virus that was isolated and identified was by a Russian scientist, Ivanovsky, and he discovered the first viral disease, which happened to be a disease of plants caused by the tobacco mosaic virus. And to the left you can see a leaf and what this virus does to the plant. The first animal virus was discovered by Beijerinck, and he discovered the virus that causes foot and mouth disease in animals. And here's a calf with foot and mouth, so you see the lesions on the tongue. In 1900, Walter Reed proved that the yellow fever virus was caused by mosquitoes. One of the most astounding things I discovered in reading and looking at documentaries on diseases was up until 1900, no scientists believed that human diseases could be carried by animals of a lower life form. There was none of them believed that flies or mosquitoes or ticks or fleas could ever carry a disease for humans. The human disease was too sophisticated to be carried by lower forms of animal life. Well, it turns out that was not true, and Walter Reed proved that mosquitoes could carry yellow fever. This was instrumental for finishing the work of building the Panama Canal, and for his work, he was an Army, here's a picture of him, he was in the Army Medical Corps, And for his astounding achievement, they named the military hospital in Washington, D.C. after him, and you probably heard of the Walter Reed Hospital. Here's the mosquito biting a person. Don't know if they're carrying a virus, but that's the vector. In 1902, Landsteiner described the 4 blood groups. He noticed that you could take bloods from certain individuals and you could mix them with other individuals, and sometimes they would mix nicely and other times they would clump or coagulate. And using this, he determined there were 4 blood groups. There was A, B, AB, and O. Here's Landsteiner. And he was awarded a Nobel Prize in 1930 for his work with the major blood types. Another way disease can manifest itself is not through a virus or bacteria, but through toxins. But up until the 1890s, none of these toxins had been discovered. Well, a Japanese scientist named Kitasato discovered the toxin that is responsible for the disease tetanus. He also discovered the tetanus bacillus. This is a picture of Kitasato probably making a vaccine using a horse. In 1890, von Behring not only discovered that the disease diphtheria was caused by a toxin, but he took the toxin and developed an antitoxin against it in animals, which they would use to treat children who developed the disease diphtheria. Here's the organism that causes diphtheria. It's a club-shaped bacteria called Corynebacterium diphtheriae. Notice that the name of the disease and the species are similar. And he was given the very, very first Nobel Prize for medicine in 1901 for his discovery of the toxin and developing the antitoxin to save many children's lives. I couldn't end the Golden Age without mentioning the syphilis spirochete. 1905, 2 German scientists, Schouten and Hofmann, discovered the organism that causes syphilis. It's a spirochete. Here's a dark field micrograph showing the spiral-shaped bacteria. In 1910, Paul Ehrlich, the same gentleman who had the humoral theory of immunity, developed a drug to treat syphilis called Salvarsan. It was a chemical made of an arsenic drug, and it was the only thing they had to treat syphilis. The syphilis in those days, if you didn't treat them, eventually it could become neurological and you would die of neurosyphilis. You could also develop heart problems, problems with the aorta, a myriad of symptoms. So this was the only drug they had. The sad thing is arsenic is a poison, and sometimes the drug is worse than the disease. One side note is in the early 20s, some doctors tried to treat syphilis with malaria. They noticed if they gave patients with malaria would develop a very, very high fever, and this fever would sterilize them of syphilis. However, then the problem was that the patient had to deal with malaria the rest of their life, so it wasn't a very good trade-off.